So the next talk will be about reconstructing biological molecules with the help of video gamers. And this presentation will be given by Robbie Joosten and Scott Horowitz. Robbie is from the Netherlands Cancer Institute. And he, among his many projects, is the PDB-REDO. He's both a bioinformatician and a structural biologist. Scott Horowitz is a professor at the University of Denver, and with an interest in, well, obviously we're going to be talking about Foldit, which is a lovely outreach to non-scientists to get them interested in structural biology that I think was mentioned yesterday, if I remember. And his lab also researches the role of RNA in homeostasis and protein folding. And so I will hand it over to them to teach us more.
Hello, everybody. Can you hear me? Hopefully, yes. So thank you for the invite for us to talk today. Robbie, you can go straight to the next slide. This talk starts actually in about 2014. I was about halfway through my postdoc, and I wasn't working on this type of stuff at all. But I was sitting at my desk, and my postdoc advisor came up to my desk and said, "Scott, I want to go skiing in the middle of the semester. Would you mind teaching my class for a few weeks?" And I said, "Sure, I can do that. What is it on?" He said, "Well, I usually have somebody else teach the NMR and crystallography parts. Can you do that?" And I was like, "Sure, no problem." And then he said, "So if you do the crystallography part, we usually give an assignment." I said, "Okay, what's the assignment?" And he said, "Well, we give the students a PDB file and, have them open it up in PyMol and measure some distances and describe what they see." And I said, "Okay, that sounds like a really boring assignment. Would you mind if I gave a different one?" And he said, "Sure, do what you want. I'm going skiing. I'll see you in a few weeks." And so he left, and I taught his class. And so the assignment that I came up with, and Rob, you can go to the next slide at this point, is basically I downloaded an electron density map, and I put in the N-terminal amino acid, and I told the students, "Go finish it." And I taught them how to use Coot. I gave them the crash course in Coot, and they went and did that. And so the first thing I want to mention about this is that the students' feedback to me was that they had a huge amount of assignment. Students were telling me that this was like a game, that they were so obsessed with it that they stopped doing all their other homework. Even though they knew that they were pretty much done with this, they would keep going back to it to try and get it a little bit better, even though that they knew it was already fine or pretty much fine. And so they really liked it. But when it came time to grading, I was like, how do I grade this thing? So I graded it the way I would usually look at crystal structures. So I ran it through a number of, computational tools. So Rob, you can go to the next slide. And so this is, of course, one of them, mole probability. And it's the histogram of the students' mole probability scores. And what's in the green line is the mole probability score of the original crystal structure. And it turned out, when I looked at about everything, I would say about 10% of the students in total had done a better job of building protein than the original crystallographers who had built it and published it and uploaded it to the PDB. They caught some mistakes that the original crystallographers had made. And so when I saw this, this immediately made me think of Foldit. So Robbie, you can go to the next slide. Foldit is, as you probably all know, a biochemistry video game in which you have many different people playing the same puzzle, trying to optimize your structure or whatever it is you're trying to do. It's graded by the Rosetta force field as you go in real time. And so you get immediate feedback as to whether what you're doing is reasonable or not. And it was originally designed for protein folding purposes, but now you can do a whole slew of different types of biochemistry puzzles in Foldit. And so we contacted the Foldit developers and asked them, do you have any interest in crystallography? Because, in my class, it seems like there's something to crowdsourcing crystallography. And in fact, they said yes, and you can go to the next slide. They had just put the features into Foldit to make electron density maps possible. Um, and they had no idea how to test whether this was going to work or not. So I said, I can help you with that. And you can go to the next slide. When the class came around again, and I taught part of it, I put together a competition, a model building competition. And so there were 61 students in my class. They were using Coot, several hundred Foldit players using Foldit. I recruited 2 experienced crystallographers to use Coot, and then I tried a bunch of algorithms. And the question was, who's going to give us the best model out of all of this? I want to go to the next slide. We can see quickly the results. Overall, the Foldit players did the best. And you can see that if you scan through the metrics here, it's not entirely clear what was happening at this point, but what we did notice immediately was Ramachandran scores was great and Clash score was great. For example, everything else was not incredibly different. So the Foldit players managed to seemingly do better in certain geometric aspects, and then but still managed to get a very good fit. In fact, still the best fit by a little bit. Okay, so if you're curious, and you can go on to the next slide, Robbie. This also, being able to load maps into Foldit means that you can do cryo-EM similarly to how you can do crystallography. And so if you go to the next slide, we did a competition here as well to see what was going on. And of course, probably most of you in the audience are well familiar with that there are many not great cryo-EM structures out there. Probably on a percentage basis, it's a bigger problem than crystal structures. And there's a variety of reasons why that's the case. And so we figured that this was an area of particular need. And so we asked whether the Foldit players could do better. So if you go to the next slide, I can outline who was doing this particular competition. It was, we had 4 subunits, so we actually did 4 separate puzzles for this one. And then we had several hundred Foldit players. We had 1 experienced microscopist using Coot and Phoenix Refine. And then I have, of course, a bunch of automated model building algorithms. And so if you want to go to the next slide again, overall takeaway from this, from an initial 1,000-foot view, was that the hand modeling was still clearly better. This was in 2019, so of course, things change between now and then. But both hand modelings were still quite, considerably better. And from a outside long distance perspective, you wouldn't really be able to tell much of a difference between them. You really have to kind of look at the details to start seeing it. So if you go to the next slide, we have a zoom in, just comparing that, when you look at the individual details of the individual residues, you can see that they don't all fit quite the same, that they've got, different rotamers, that you can see the backbone is slightly different in many cases. In this case, I'm just showing one of the automated algorithms. This is the Phoenix, and you can see that it in registry issues. So it was having much larger problems than the humans were. But when you look at just these details like this, you're not really sure whether the Foldit players or the microscopists did better. And so for that, again, you have to dig into the stats. So if you go to the next slide, Robbie, and we can run through these pretty quickly. Clash score, Foldit was definitely the best by a wide margin over the microscopist. We go to the next one. That's Ramachandran, which of course, once again, the Foldit players did a much, much better job with. And then one more time, we will see the fit to density. And in this case, the Foldit players really, it was pretty much an insignificant change for the microscopist. So putting this all together, essentially the folded players were a bit more conservative in their model blend, which I should mention this is cryo-EM, so there's no iterative refinement here. And so in essence, the best folded solution is the best. And we've actually run many other tests now to show that the thing that you get in a cryo-EM is the best folded score is really good, darn good structure. And so this tells us that yes, you can use Foldit for cryo-EM puzzles, and they could be extremely useful for the community there as well. Okay, so continuing on, you can go to the next slide. I started thinking about whether we could improve already existing structures in the PDB using Foldit puzzles. This seemed like an obvious thing to try. So, I grabbed an example, one FHE, and if you hit the next one, you'll see the stats for it. And obviously, this is not a crystal structure to write home to your parents about in terms of your model building. And it's got a lot of obvious problems. And so I made a puzzle out of this and gave it to the Foldit players. And I figured that the best way to compare this to see if it was useful was to compare how the Foldit players did versus PDB-REDO. The gold standard for doing this. So this is the original PDB and the original PDB-REDO entry for this particular protein. And you can see that PDB-REDO does, improve it considerably. But it's, still not, you're still not looking at this like, really, is this good? The Ramachandran plot is still really poor. It's better, but it's still really poor. And of course, the R factors are not exactly amazing either. So I took the result from the Foldit puzzle, found the best Foldit puzzle solution, and then ran that through PDB-REDO as the starting place for PDB-REDO. And then this is what you get out of that. The Ramachandran plot is still a bit messy, but everything else improves quite a bit. And even the Ramachandran plot does improve a little bit. And so this told me, hey, there might be something to this. Maybe we should be funneling the worst PDBs through Foldit players and then potentially to PDB-REDO after that. And so I ran a few more of these puzzles and saw similar things. And so at that point, contacted Robbie and was like, hey, we think we might have something useful here. Are you interested?
Yeah, and indeed, I was very much interested. So following up, what we really wanted to do is then get these Foldit puzzle results and make them available through the PDB-REDO databank, but not as a final model, but rather as an input model, so that it still goes through the PDB-REDO procedure and users get what they expect from PDB-REDO, only hopefully better. And because Scott and I wanted to have it maintenance-free, we made sure that everything would be fully automated. And of course, because it's a data bank, everything has to be fair, and we have a clear provenance of where the model comes from. And of course, this is a model made by somebody else than the depositor, so that person, the Foldit player, should also get some sort of credit. Now, to get these models into PDB-REDO, there's a few things that we need to consider. First of all, there's not just one Foldit solution, there's hundreds of thousands of these Foldit players make lots of alternative models, and then you have to figure out which one is the winner. So what they did is the models are clustered by just C alpha trace, and then from each cluster you take the one that has the best Foldit score, which essentially comes from the Rosetta force field. And those models, cluster leaders, call them, are run through the PDB-REDO API, and it's the same thing that you have straight from CCP4, and we just do model refinement. So no rebuilding, we take the sort of build state from Foldit, and then from the results, we take the one with the lowest R3. So now we have a sort of defined Foldit winner. But then the next question is the winning solution actually behaving better in refinement than the original PDB model? And to figure that out, we did a bit of a benchmark. We took at that point existing 85 Foldit models, and we ran them through PDB-REDO. So now we're now comparing refinement, PDB-REDO refinement of the PDB model on the x-axis and PDB-REDO refinement of the Foldit model. And if you look at the distribution of R3, essentially, changes are not that big. There's a few cases where things become significantly better or significantly worse, but the majority of the cases is around the diagonal. However, if you look at the geometric scores, you can see that the Raman plot improves a lot. For the majority of cases, same goes for hydrogen bond satisfaction, fine packing, so that's the details of how the atoms touch each other, and also the mole probability score improves most of the time. So essentially, we can say that there is a very poor correlation between the data fits and the geometric quality. So now we have to deal with that. Of course, for the PDB-REDO data bank, we want to avoid making things worse, particularly in terms of fit with the experimental data. And also, we want to avoid running everything twice. And as James already pointed out, there was always particularly when you're almost done with the model, there's a trade-off between good model geometry and fit to the experimental data. And we know for the Foldit models that they have typically have better geometry than the PDB models. So to deal with this, we do a trick that we do a lot in PDB-REDO. We do a comparative refinement where we change only one parameter. And in this case, the parameter is the input model. So we do a quick refinement in RefMaCat, auto-weighting all the extra restraints that PDB-REDO generates, and then see which model performs best. And in this case, we have a bit of a biased selection. So we take the Foldit model unless it has a much worse fit to the experimental data. So if R3 goes up with 1 sigma or the LL3 goes up, in that case, we take the PDB model as input, but otherwise we take the Foldit model. So this is purposely biased to improve the geometry. Now, if you apply this algorithm, and now we're comparing refinement of the model with PDB input or the one that is selected by the PDB-REDO procedure, you can see that there's fewer cases where R3 gets worse, but still we have a lot of cases where the geometry improves. So there is, of course, some loss in geometric improvement, but we're now safe by not messing things up in terms of fit of the experimental data. So this approach works, but now we have to do something else. We have to work on the automation to make sure that this will continue working for the future. So we have to future-proof it. And what we did is there's a new API for the Foldit server. So now every week, the PDB-REDO server talks to the Foldit server through a secure connection and asks, are there any new Foldit solutions? And if there's any Foldit solutions for a particular PDB entry, then this PDB-REDO, the related PDB-REDO entry is marked for updating. And then with the weekly update cycle, it gets replaced. So, and in this update cycle, we do this test again. We see whether the original model or the Foldit model works best. And if the Foldit model works best, obviously this one goes through to PDB-REDO to the end result, but we also make sure that we capture this in all the metadata. And it's these metadata that are fully documented, of course, but you're allowed to, you can search these and you can create data sets for it. So we have a thing called the PDB-REDO Archive Manager, where you can just look for different parameters. In this case, here on the right, you see, is Foldit used or not? And you can see that there's 83 cases where the Foldit model is used. But you can also filter by R3. You can filter whether or not by PDB-REDO thinks your data are twin. But this is a general tool that you can use to make data sets for any sort of exploratory research. But moving back to the PDB-REDO data bank, this is now what it looks like. So now if you have an entry that's clearly marked that it's done with PDB-REDO version this and this, with the Foldit model, with a particular model number, and this model was made by, in this case, player Bruno Kerstemont. We're using the PDB model. It says the exact version of the PDB-REDO model, the PDB model that we used, because PDB models get updated as well. So if you look at the refinement results, these were the original ones, and you can see that they were not great. In most cases, the geometry actually gets slightly worse, and the overall scores are pretty bad. But for this entry to FX2, with the Foldit model as input, you can see that R3 improves a lot. So it goes down with like 4.5 percentage points, which is really a lot. And all the geometry scores improve, actually a staggering amount. But of course, these are just numbers. So let's have a look at the structure. And what you can see on the left here is the original PDB structure model, I should say. And on the right, the thing that comes out of Foldit and then PDB-REDO. And you can see there's changes in the secondary structure, particularly the central beta sheet becomes much more elaborate. You lose some beta hairpin on the top, but the important part is actually in this loop, because this is a surface loop. This is important for interactions. And if we zoom in here with the electron density with it, you can see that we had a register error. So, and this register shift is around this glycine 411. And this glycine is a bit weird because it's fully conserved over 1,500 sequences. So apparently it's very important. And now this is a yeast protein, but if you look at the human equivalent, then you can use alpha missense very conveniently. And you can see that any mutation of this glycine is deleterious. So apparently this is a key residue. So what we show here is that with these essentially folded puzzles, with these games, you can still change the biological interpretation of a structure model. And yeah, that's pretty much what we want to conclude. So human input can still sometimes trump automation, which I think is a good thing, particularly in the age of AI, where everybody's like, computers should do everything for us. What it also shows is that the wisdom of crowds, completely disregarding everything what happened during the COVID crisis, actually still exists. And more general, that revisiting existing structure models is something that Vladek said yesterday as well. It keeps them useful and informative for the future. So really, if you can update your PDB entry for something better, then you should do that. And finally, Scott has informed me that we take requests. So if you have a particular PDB entry, say, well, can you put that in the front of the queue for a folded puzzle? Then we can consider that. And it's not just protein, but also nucleic acid. So with that, I'd like to thank a few people from the University of Massachusetts in Dartmouth, Firas Khatib and Andreas Petrides. And at the side of the Netherlands Cancer Institute, Martin Heckelman, who did all the updates to the PDB-REDO website, and Anastasis Parakis, who's of course the boss here. And I also want to thank our research high performance computing facility, because we provide a resource as a cancer institute. This is not our primary research, but to do these sort of things, you need an infrastructure. So having a facility that makes sure that your servers are maintained and everything is running so we can do the science is very important. But I think the most important is the folded players in this case, and of course also our funding bodies. Now, if you want to try this for yourself, which I highly recommend, you can follow the top QR code, go to Foldit, do one of these puzzles, it's fun. If you just want to look at the results, you can also go to PDB-REDO from the bottom QR code. And with that, I'd like to thank you, and I hope there's still time for questions.
So there is indeed time for questions. See a couple of hands going up. We also have a question online. How do you make a folded puzzle? Is it quite an involved process?
I guess I should answer that. So not really, if you know a thing or two. Basically, it's a very simple JSON puzzle setup file and your PDB, and then you can upload it. You have to have permission to post puzzles, but you can also get permission to post private puzzles, so you can run your own little puzzles if you want. And that you know, is much you can kind of play around with that a lot before you try posting a main folded puzzle. So we've had people do that before as a way to get introduced. But yeah, it's a very simple JSON file with with your parameters, and you can get really fancy with it, if you're adding a bunch of restraints and such. But at the basic, you can just give it a PDB file. The biggest issue is that you have to know which types of ligand understands and which ones they need parameter files for. And so you can actually look that up within the Foldit files itself. And then if you don't have one, then you might have to search for a Rosetta parameters file or make one. And if you have to make one, that's actually the thing that's hardest, and takes a little bit of Rosetta acumen to make happen. But the way I get around it is I basically don't post puzzles with ligands, except in dire circumstances. And so if you have really essential ligands, you'll have to go through it. But otherwise, you oftentimes get away with it and then just plug the ligand bit back in at the end once your model is considerably improved.
Yeah, Robbie, Randy Reed here. So getting to the standoff between humans, the wisdom of crowds and automation, what happens in this case where you've got the register shift if you look at an AlphaFold 2 model? Because they're often, the register shifts are really rare in those models.
Well, we looked at a different case where there was not a register shift, but rather a loop doing a big movement that looked like a register shift if you look at the details. And there, the AlphaFold model was sort of in between. So you had the folded model on the one side, you've got the PDB model on the other side, and the AlphaFold model was somewhere in the middle. That was very poor density. So if you look at the overall fit with the data, I would go for the folded model. But that case was particularly hard to prove that the AlphaFold model was worse or better. But the statistics overall would favor the folded model.
Okay, I have comment and question. One comment is that the main difference between PDB and PDB-REDO is that you may correct structures and PDB cannot. Yes, PDB is a repository of what people are putting in. My first question is, when you will find serious problems in a structure, why you do not alert people?
I do. I always, if there's serious problems in particular structures, particularly when it's about the data, I admit, I contact the depositors and the PDB annotators at the same time so that the errors can be corrected. And we have a long track record of fixes in PDB entries based on the feedback that we provide as PDB-REDO. We also have there's a nice case of a really biologically error in a PDB entry. It was a long time ago, but there was a protein peptides structure model, and it was clear that there was a peptide flip in the peptide, and it made no sense. We contacted the authors, because at that point you still had to replace a PDB entry, withdrew the PDB entry, and then uploaded the PDB-REDO model as the new PDB entry. So yeah, we go after these sort of things.
Okay, my impression was different. What is very nice that I found that you react to what people are writing to you. I alerted you about waters, and you have changed the way of handling waters, which I noticed yesterday.
Yes, if and I think that's true for all developers, right? Feedback is always very much appreciated. Sometimes things are hard to fix, admittedly, but overall, when people come with feedback, we always try to fix things. We're a small team, so it sometimes takes time, but yes, feedback is always welcome. Actually, there's something I'd like to ask as well. James, can we make your challenge into a folded puzzle? That would be interesting. And don't tell everybody what the solution is, but that could be an interesting extra test.
Right, yeah, Dan Rigdon, University of Liverpool. Does the folder infrastructure capture what the humans are actually doing in a way that would allow us to spot, little tricks that are not currently options in molecular graphics or in the automated pipelines?
We have two ways of doing this. The first is that we do log every move the folded players make. And in the first case when we did this, we did actually track carefully, what the winning team did. It's been long enough now, they don't remember the exact details, but it wasn't something that was going to be shocking to you. But it was, a combination of various, hand and automated movements in a particular order. In this most recent work, we actually reached out to the Foldit players that produced the best structures across, these first 50 plus puzzles, and asked them to give us, because these are people that, had clearly figured something out. And it was really quite different. Some people were doing a lot of hand modeling with very particular orders of things. Some people were using primarily algorithms. Some people actually did entirely algorithms, didn't do a single bit of hand modeling, and still won several puzzles. And so that tells us that there's actually quite a lot of room for improvement in automated crystallography when a Foldit player figures out a set of algorithms that can substantially improve several of these. And so that's all in the paper. These are all listed in the supplement. The biggest issue there is figuring out what the algorithms mean when they name them, because you actually have to do some Foldit digging to find that in many cases.
Interesting, thanks.
Hi, Paul Bond from University of York. The thing you said about preferring the Foldit model seemed slightly worrying. So that if the two models were the one from the depositors and the PDB, and the one from Foldit, if Foldit was the same or slightly worse, you would pick that to go into PDB-REDO, or is the answer that you're just taking both? So you're presenting the results of PDB-REDO on both of them?
No, we test both of them. And so for all the puzzles that we have now, and the puzzles are biased towards structures in the PDB that have very bad statistics. So that's why we can apply this trick. So in those cases, at least at the input stage, the Foldit model always has much better geometry, but sometimes worse fit with the experimental data. And that then in refinement, if in refinement the fit with the experimental data stays worse, then we use the PDB model. So it is biased a bit, but I think that the bias in this case you can get away with because of the selection of the Foldit model. If you would now throw very good structure models into a Foldit puzzle, then we have to change the algorithm. So it is a limitation, but it's by design.
One more question.
Hi, Robbie. Dave Briggs, Francis Crick Institute. This follows on from Dan a little bit. How much of the improvements are down to sort of the Rosetta refinement engine, and how much do you think of it are down to genuinely the wisdom of crowds?
Yeah, we get this question a lot. And this is why we always actually run, in our benchmarks, we run Rosetta. It's kind of similar functions for this. And we've never seen Rosetta do anywhere near as well as Foldit. And so there's clearly a human-computer interaction component that's really important. I do think that the Rosetta force field has some real strength that humans are able to work well with, and can certainly help with things like, I think a lot of the Ramachandran and Clash score improvements do come from Rosetta, but Rosetta itself can never manage to get to a position where that's really useful. And so it's really the interaction that seems to be important.
Okay, a final round of applause for the last speakers of the session. Thank you very much.